Hello, my name is Mac Merritt. I'm a senior at Georgia Tech, and I've done undergraduate research for a while with fMRI, and as a project for my senior year in neuroscience, I've decided to create this video on speaking the language of fMRI and functional connectivity. When I first joined my lab and they started talking about all the complicated terms involved with fMRI and functional connectivity, I had no idea what was going on, and sometimes I was a little afraid to ask questions. So I wanted to create this as just an overview that can kind of give you an idea of the whole process of what's going on, and then you can dig more deeply into different areas as you have interest. I created this because it's really confusing what's going on. You start getting a whole bunch of terms thrown at you, like B naught, RF field, nuclear spins, everything ranging from quantum physics to signal processing to gradient coils. There seems to be so many different things that people are talking about, so many different fields that come together to make this field happen, that it's almost impossible for an undergraduate researcher to be able to understand it all. So hopefully by the end of listening to this video, you'll be able to understand a little bit of what people are saying, and you may not fully get the depth of it, but you can at least know where to look from here and be able to trace everything back to a big picture. So one of my main goals is to continually go back to the big picture of what we're looking at with all of these nitty-gritty details. As an overview, I'm going to start out with some background on MRI in general, being able to understand where the images come from, because a lot of times people are referencing some of the physics. This is kind of a quantum physics section, and I don't really know quantum physics well, but again, just describing an overview of the language. Then I'm going to go into some details of how functional MRI works and how that's distinct from traditional MRI. Some of the traditional methodologies that people use, such as block design or event design, how they analyze those, and what kind of steps go into pre-processing the data. I'm going to go into functional connectivity after that and resting state MRI and how those are related to traditional things. So starting with MRI in general. MRI works, you probably heard it referenced as a really big magnet, and that's essentially what an MRI scanner is in its most basic form. The coils create a really strong magnet in the center that's on the scale of 3 Tesla, maybe down to 1.5 Tesla. Some animal scanners get up to 9 Tesla. And that word Tesla is just a term used to describe the strength of a magnetic field. So that magnetic field, the B naught field, is the strongest magnetic field in the scanner. And it essentially aligns the water molecules inside of someone. And it does this because there's a charge spinning continually. It creates a magnetic field. And this magnetic field is pointing in some random direction, like on the graph on the left there, the B naught equals 0. And then when B naught is applied, they all align in the same direction. This is really useful because if you apply a radio frequency pulse to it, it knocks it out of alignment. And then you can measure how fast it returns to alignment. And this measurement is what generally gets you the MRI image. And you can look at a couple different ways that it returns to baseline. There's what's called a T1, a T2, and a T2 star. And these are different relaxation times to take to get back to that baseline that you can look at. And they have different properties. From a clinical perspective, they give you slightly different contrast in the images. Different tissues are going to light up slightly differently. So the benefit of all of this, the reason that you want to be able to fire a pulse into a scanner and the reason you want to be able to see how fast it returns is that different tissues have different relaxation times. So, for example, gray matter in the brain has a different relaxation time than white matter, which has a different relaxation time than cerebrospinal fluid, which has a different relaxation time than a tumor. So if you were to scan someone's brain, you could see a distinction between all these different types of tissues, see that there's a tumor in the brain, or see that their gray matter is abnormally high. The only thing we're missing at this point, though, is all we've gotten back is a single stream of data from a radio frequency scanner. And there's no way to interpret whether or not a given scan is from one side of your brain, the other side of your brain, or from the top or the bottom. We have no spatial information. And that's what the gradient is for. You may have heard of a gradient. And what it does is it applies a change to the field slightly. And then you can see a general linear trend in your data. And when you interpret that out, it gives you spatial coordinates. And that may be a little bit complicated to understand, but basically the idea is that a gradient coil is a big coil built into the scanner that allows you to be able to see different spatial information about your data. The main idea of MRI as a whole. is that a person enters a scanner and the MRI takes a picture of their insides, essentially. And you get different contrast for different tissues that are outputted into a three-dimensional image. It has pixels in one direction. If you took a single slice, people often call it, you could look at it just like a picture. People usually call the individual three-dimensional pixels voxels or volume pixels. So if you hear somebody reference voxels, that's usually what they're referring to. So then, if you imagine that we took an MRI scan of someone's brain, and then we took another one immediately afterwards, and another one immediately after that, as we do that more and more, we enter into the realm of what's called functional MRI. And it's basically just MRI with a time component. It's like a movie of MRI images. And we use a different type of scanning sequence called echoplanar imaging, which is much faster but much lower resolution. So you can see the image there on the right. It's kind of blurry because it's been acquired with echoplanar imaging. And the benefit of doing this is that just like different tissues give different contrasts in an MRI image, different blood oxygenation level of the brain gets different contrasts. So if the blood has more oxygen in it, it's going to give a different contrast level than if it had less oxygen in it. Why? And why is this valuable? Well, as neurons fire, they need more energy. And so there's going to be more oxygenated blood flowing to the regions with greater neural activity. This causes the contrast in those regions to increase, and so you can get a rough estimate of the neuronal activity in a region based on the contrast of an fMRI image. So if a pixel or a voxel gets brighter, you could potentially say that there's more neural activity in that region. Keep in mind that this is an indirect measure. So the way that people make sure that they're not missing something or getting too much noise is often they'll use an electrical validation. They'll use electrophysiology in animals where they implant electrodes, or they'll use EEG in humans. And this helps us to be able to interpret the fMRI signal. It's also how we know what the fMRI signal is measuring in the first place. And you hear a lot of people when they compare fMRI data to the underlying electrical and neuronal signals, they'll use a term called band-limited power, or just BLP for short. This is a little bit difficult to characterize without some signal processing knowledge, but as a rough estimate, you can break down a signal into different frequency components. You can say, I'm only going to look at the fast changes, or I'm only going to look at the slow changes. And then fMRI is related to how the amplitude of those frequency bands changes. So if you looked only at the high frequency data, you could say that fMRI bold data, blood oxygenation level dependent signal, is related to how the amplitude of the high frequency data is changing in time. This may be getting kind of confusing, kind of wordy, depending on the background knowledge you have, but as a main idea to keep track of what's going on here, if an fMRI person still physically enters the scanner, and the MRI scanner scans their brain and outputs electrical signals. This time, it's going to output a bunch of signals acquired in time, so you have a movie of three-dimensional images. So your data is four-dimensional, just like if you were to look at a regular movie by itself, it would be three-dimensional, because you have 2D plus time. You have 3D plus time, and then you can look at the brightness of each of the voxels, the volume pixels, and be able to get a rough estimate of what's going on with the neurons, because that's the blood oxygenation dependent signal. So now we're going to get into some of the fMRI methodologies, how it's done. To start with, we need to go over preprocessing. You have that three-dimensional movie playing in time, and the signal looks completely useless, just a bunch of noise. So what do you do with it? The first thing you have to do is pre-process it. Pre-processing is a pretty complicated process. You'll typically use a software like SPM or AFNI, and you'll do things like motion correction, slice timing correction, spatial filtering. You've probably heard blurring referred to, time filtering, and global signal regression and registration. These are all really complicated terms that essentially are getting the noise down or the signal up. You're trying to increase your signal to noise ratios. You have some useful stuff and some just completely useless information in your signal. fMRI outputs a lot of data, and so you have to be able to weed through it before you can actually use it. The basic way we're weeding through it is by first correcting for motion. So if somebody moves around in the scanner, it looks like all the pixels of the brain have changed. But we can correct that by knowing that the edges of the brain change. That's what motion correction is. And all the other things are related to filtering. Filtering kind of goes back to that signal processing information we were talking about. But basically it gets rid of really sharp changes in either the spatial information, so like if one voxel looks super different than the voxel right next to it, that's probably not really meaningful because most regions are gradual shifts. So we just kind of lessen that sharpness. And the same thing in time. If something changes really quickly, then we can lessen it, or if something changes really slowly because of a gradual drift, we can lessen the effects of that. And there's two different traditional ways to do fMRI. A block design is where you give them a task in blocks. You might say a minute of doing some task, a minute of not doing a task, a minute of doing some task, a minute of not. And then you look at the differences in the fMRI signal during the task and not during the task. So let's say you just had somebody tap their finger for a minute and then not tap their finger for 30 seconds. And then you looked at the signal, you average the signal in the motor cortex. You might get a data like this, where the red line represents when they're told to tap their finger, and the black line represents the average bold signal of their motor cortex. And you can see it's higher during that time, so you could say that that area is active. The bold signal is heightened during that time for that task. The second way is an event design, which is a little bit more complicated but has some benefits to it. So an event design involves giving a single event and then seeing how the brain responds to that. This allows you to be able to distinguish between subtle differences, but the downside is that you have to be able to estimate how the signal is going to respond according to a model. Remember that bold signal data is not a direct measure of neural activity, so you can't just say it's going to respond with the neurons. It has a slow hemodynamic response, meaning blood flow response. That's a delay from the actual signal. So we compare our data to a model of what the data would theoretically look like and see how closely it matches that model. And that gives a rough idea of how much the brain is responding to that task. And this gives you distinction between individual tasks rather than just block sets of tasks. For typical fMRI analysis, you use a general linear model to be able to analyze the data. And a general linear model essentially divides your data into components that make it up. It says that it's going to be made up partly by error and partly by interesting things. This X component here is things that we know something about. So it may be just low frequency shift, like a general trend over the course of the scanner session, or it may be when your motion occurred, or it may be when your task occurred, which is the most meaningful. And then the beta value is going to tell you how the fMRI signal in individual voxels changed at the time points that you're looking at in your X matrix. So this takes in your fMRI data and information about what happened during the scanner session, and it outputs how your task and how other things that happened impacted different regions of the brain. Back to the main idea. fMRI methodology consists of the same steps starting with fMRI, and then once you have your data outputted, you're going to do post-analysis on it to find out meaningful information about the brain. This post-analysis consists of two main steps. Pre-processing is where you get rid of some of the noise and try to make the data into a more useful format. And this is going to depend on the characteristics of your data, what type of pre-processing steps you're going to do. But if you're interested in it, there are really great videos on how to do it. And then analyzing the data according to whatever method and whatever test you're trying to do, often using a general linear model, but sometimes using other types of analysis tools. Again, many videos on this in more depth. Alright, finally to the topic that I'm most interested in and what my research has actually been about is resting state fMRI and functional connectivity. Resting state fMRI basically just means someone sitting in the scanner doing nothing while not sleeping. Not to be confused with sleeping, it's just resting, just sitting there. This is used to analyze functional networks often, and it's looking at spontaneous fluctuations. There's no task going on, there's no stimulus, and it makes it very difficult to analyze because people's brain activity is no longer time locked. You can't compare how one person responds to a stimulus to how another person responds to a stimulus because there isn't a stimulus. And so instead we compare each individual brain signal from different regions to itself or to other regions in the brain. And this gives us what are called functional networks or functional connectivity. And you can see in this picture many different functional networks that have been found. It's kind of difficult to interpret them because all this is really saying is that the regions that are lit up here are statistically more likely to occur together than to occur opposite or differently. And what I mean by occur together is activate in a similar time manner. So this isn't necessarily telling you something about the electrical or the neural activity, but there's a lot of research to investigate what these networks actually mean and how related they are to neural signals. There's two main ways of doing this. The first is seed-based correlation. The way this works is you pick a seed, like the one in the top left, which would be the somatosensory cortex, and then you look at how other portions of the brain are related to that seed signal. So in this case, if you took the average signal of the somatosensory cortex, then you would look at all the other regions of the brain and see how correlated they are to that seed-based signal. And this result that's seen in the top here is pretty typical, where you have high levels of correlation from the bilateral version of that. So the somatosensory cortex on the other side of the brain is highly related to the one on this side of the brain. One of the other common methods of doing functional network analysis with resting state fMRI is using independent component analysis. Independent component analysis is a signal processing tool that allows you to be able to separate independent components based on the name. It's typically described in terms of a party problem, where you have multiple voices happening or multiple conversations happening, and if you have enough microphones in the room, you can separate them, like the example here where both microphones are recording data from both Kyle and Stan, but using independent component analysis, you can separate the independent voices. This is related to the way that it's applied to fMRI, where the theory is that different regions of the brain are having different conversations. And because we have enough information from the scanner, we're able to divide it into the different conversations, components, or different functional networks, and output each of those. These tend to look very similar to seed-based correlation generated networks, but the difference is that these are hypothesis independent, meaning that you're not picking a region that you're interested in, you're just saying what in the brain is interesting as a whole. And it is interesting that they tend to look similar to the seed-based correlation. So, getting back to the main idea finally for resting state fMRI and functional connectivity. Person enters the scanner still, outputs a 3D image with time, 3D movie. And then instead of doing a typical post-analysis, you're still going to do pre-processing, although your characteristics of the pre-processing is a little bit different. People might refer to it as lower frequency because that's typically what the information people are interested in from functional connectivity. But you're going to output that video, three-dimensional movie, and you're going to compare a brain to itself. You're going to compare one region of one brain to one region of another brain, or all the regions of one brain to all the regions of that same brain. And this gets past the time-locked issue because each brain is time-locked with itself. As a conclusion, functional connectivity is quite complex, and so I encourage you to ask questions. If you're entering a research lab, often professors are very eager to explain what they do to new incoming students and to be able to help you develop the skills to be a great researcher. And in developing those skills, it's great if you can develop an interest and a passion for the thing that you're investigating, because you'll tend to learn more about it, be a more effective researcher, and be more knowledgeable about the topic because you'll want to learn the things. If you're considering what topics to study that might be helpful for understanding some more of the language, the first thing I would say is signal processing is essential. Image processing and time signal processing are used all the time in fMRI. And if you can take a class on DSP or you can just learn about it on your own, it's going to help you in innumerable ways. I find that many of the coding that happens with fMRI and the analysis occurs in MATLAB, so if you can acquire that coding skill, it's pretty helpful. And then typical engineering skills like linear algebra and statistics are great. If you want to understand more of the physics behind it, studying quantum physics, nuclear physics are valuable for that. I don't have a great knowledge base on that, but it's good if you're going to be altering pulse sequences or changing the way that you're doing fMRI rather than just analyzing the data. Thank you. This is a list of links that I used for all the pictures that you've seen. All of them were taken from openly available resources online, Wikimedia, for example.